Okay. Um, Second to last video of this unit. The last one should be a relatively short one, but this one is a big topic. It might not be a super long video, but we're going to spend a good amount of time with it in class because it makes up a good portion of this unit. The name of the topic is chi-square analysis. Do not say chi-square or chi-square. I will lose it. It is chi-square. At least I hope so. And it's a statistical analysis we do basically to try and determine if genes are following Mendelian inheritance rules or not. So I've titled it as AKA When Genetic Crosses Don't Look Right. So we've kind of looked, we've done our Punnett squares and we have like the 3-to-1 ratio, the 9-3-3-1 ratio. So this kind of deals with what do you do when your data doesn't fit either of those ratios. So we have a formula. You don't need to memorize it. You don't need to memorize what any of the variables mean. It's all on the equation sheet. But you do need to know how to use it, which we'll get into. But again, the main purpose is to just determine if the data is good or non-biased, or if it's bad / biased. Meaning, am I just unlucky with my data, or is there actually something else going on? Like if I flip a coin 10 times and I get 10 heads in a row, this chi-square analysis basically lets you put some math to like, okay, what are the odds that I actually got 10 heads in a row, or is it more likely that there's something up with the coin and it's not actually 50-50? So when I say bias, I just mean data being different from what you expected. It's biased in a direction. And there's something causing the difference. This analysis doesn't tell you what that cause is. It just tells you that it's probably not random chance that caused you to get 10 heads in a row. It's probably something else. Could be the coin, could be how you flip it, doesn't matter. So the basic formula, we get the chi-squared. You don't do anything with this Greek letter chi. This is why it's called chi-square analysis. The main stuff is over on the right. So this fancy E means a sum. We have to add together a bunch of different terms. So you might have a data table with four sets, 4 categories for each different phenotype. And so you'd have to do this equation 4 times and add them all together at the end. And what you do is you count up the number of observed individuals you have with a given phenotype, and you subtract from it the expected number of individuals. Square that, divide it by the expected. It gets you one of your terms. You do that, repeat that for each of your data points, add them all up, and you get this chi-square letter. Okay, so before we start a chi-square analysis problem, we usually have to write down what is called a null hypothesis. So a null hypothesis is a little weird. It's different than whenever in science we've asked you to write a hypothesis. That is not what a null hypothesis is. A null hypothesis is basically just a statement that any differences you see between the observed values and the expected values are completely due to random chance, occurred by chance. There's nothing else going on. And then once we have our null hypothesis, we seek to either prove or disprove that. So if the null hypothesis is supported by our data analysis, by the chi-square analysis, that means that, yeah, mating is random, normal gene segregation, independent assortments happening. Basically, it follows Mendelian inheritance, which is the assumption in every genetic cross when we do it. If the null hypothesis is not supported, so if we disprove it, that means that the difference between what we observed and expected is big, and that something non-random is probably happening. It's if I flip a coin 10 times and get 10 heads, maybe it's random chance. If I flip a coin 1,000 times and get 1,000 heads in a row, well, maybe it's pretty obvious there's something going on there, something non-random. So possible explanations for this is just genes are not randomly segregating. They could be sex-linked. They could both be on the X chromosome. They could just both be on the same chromosome in general and inherited together. There might be more genes impacting these than what we're impacting the phenotypes than we assumed. A million explanations, you're not necessarily asked to come up with them. The other thing we're going to need before we can actually start a chi-square analysis problem is what is known as the degrees of freedom. And degrees of freedom is basically like how much wiggle room does your data have. And the general idea is that the more possibilities, like the more phenotype possibilities you have, the more random chance you're kind of allowed. If there's 10 possible outcomes, you're going to have variance if compared to just 2. So if you have just 2 possible outcomes, you're not really allowed to have much random chance, but the more possible outcomes you have, the more wiggle room you get, and the more degrees of freedom you have. The simple rule is just it's the number of phenotype groups minus 1. We do have to be careful with it. It's not just phenotype minus 1. It can get a bit more complex. We'll deal with a practice problem that kind of goes over that a bit more. But then using that degrees of freedom value, we have to head to what is called a chi-square table, which I will show you in class in the slide, in the next slide, I think. But we need to go grab a value from a table, and then we basically compare our calculated value to this value from a table, and we can draw a conclusion. So let's look at an example, because it probably is not making too much sense yet. So standard dihybrid cross, we get 2 flies, the wild type and the mutant type, which is black body eyeless. We cross them all together. The F1 generation is all wild type. So having eyes, having whatever color that is, body, is dominant. And we get these results. Need water. So get the results. Mostly we're wild type. If you look at these, whenever you have this kind of distribution where you have clearly one has the most, 2 of them are about the same, and then one has clearly the least. If you add these all up, divide it, calculate it, it's close to a 9:3:1 ratio, but it is not exactly a 9:3:1 ratio. There's a little bit of noise, a little bit of mess in it. So we're going to do a chi-square analysis and figure out, is this due to random chance, or are they linked, or something like that. So first thing we do is take a drink of water. Oh, apologize. So we need to get the total number of offspring, because we need to be able to find the expected values for the dihybrid cross. What was on the previous page was the observed values that we actually got by doing it. But we need to use the 9:3:1 ratio here to calculate the expected values, because 9/16 should have been wild type, 3/16 normal body eyeless, so on. So we use those fractions, multiply them by the total number, and you will get your expected values. Yeah, this is just saying that. Multiply, the fraction by the total number of offspring, and you get your expected number, and we can populate our table like so. So now we can directly see what technically should have happened. And we can see those numbers are pretty close, especially when we're on the order of 10,000 flies. This is almost certainly going to pass the test, but we should go through, write our null hypothesis. And again, the null hypothesis is just that any differences we see are due to random chance. So 2 traits are not linked, and therefore randomly segregate and assort independently. During gamete formation, differences between expected and observed values are due to chance alone. Really, you could shorten that to just say the 2 traits are not linked. The differences between the observed and expected values are due to chance alone. So we have a null hypothesis. Always shows up on the exam. Now let's calculate our chi-square value. So we take our observed values, subtract the expected value, square it, divide by the expected, and then we get 4 small little decimal numbers that will add up together, and we get our chi-square value is 0.3. And I'll tell you what we're going to do with that in a second. We need to find the degrees of freedom next. So how many different phenotype combinations are there? There were 4 possibilities, 4 possible outcomes. So to calculate degrees of freedom, it's just phenotype groups minus 1. And we get 3 degrees of freedom. So we're going to head to a chi-square table where we need to find, essentially, we're going to be using their 0.05 for P almost always. And so kind of what you're checking is you say, okay, 3 degrees of freedom. Ignore this red bolding. 3 degrees of freedom. We always use 0.05. This means that there's, if our number is less than this 7.82 value, that means there's a 5% chance or less that this, what we saw, is due to something else happening. So basically, if our value comes out to be less than 7.82, there's a 95% chance that our data is not biased, that it is following the rules we expect, that the changes we see are not due to anything but random chance, which is the case we fall into. So this does it a little differently. Essentially, what I want you to think is, yes, the 0.3 value we calculated is much less than 7.82, which means there's a 95% chance that our data is following Mendelian inheritance rules, and any deviations are just from random chance. So if it is above that 7.8 value, then we would reject it. So if we had calculated our chi-square value and it came out as 8 with 4 degrees of freedom, if we have a chi-square value of 8 and we have 4 degrees of freedom, do we fail to reject, or do we reject it? With, going back, 4 degrees of freedom, the answer is 9.49 that we're comparing to. And so we still fail to reject it. It had to be over 9.48 with 4 degrees of freedom. And so again, random chance was probably the reason. So you can kind of make it formulaic in terms of how you write your conclusion statements. These are very popular FRQ sections. And if you paid attention in the pre-test practice exam thing, there were a good amount of these in there. So it shows up, need to know all parts of it. But you basically just need to say, if you want to be fully correct all the time, with blank degrees of freedom, my chi-square value is whatever you calculate, which gives me a p-value between 2 percentages, and therefore you either accept or reject your null hypothesis. But you don't try to actually explain what's going on. If you need to do any further explanations, that would come in a separate study, separate area. This is just to say, yeah, there's something fishy going on. We need to look into it further. And that is chi-square. I am not going to go over linkage and gene distance in this. That is the kind of trying to explain the results of a chi-square analysis. That'll be in the next video. If you are freaking out and don't understand how chi-square analysis works, we are going to go over it in class. I'm going to re-explain it in class, and we'll do some practice. I just wanted to get you a brief exposure to it before then. I'm sorry if it was painful and 15 minutes long. I'm so sorry. Goodbye.